This class is on diseases of chickpea or Bengal gram. Chickpea, one of the major proteinaceous pulses, being utilized as a protein substitute throughout the world, and this crop is highly susceptible to many pests and diseases. Among the diseases, what are all causing major losses, we are going to see one by one in this class. The root rot, which is common in many of the pulses, caused by Rhizoctonia solani, and then wilt caused by Fusarium oxysporum, Ascochyta blight caused by Ascochyta rabi, rust caused by Euromyces ciceris, and then foot rot caused by Operculella pudwicki, and then cottony root rot or stem rot caused by Sclerotinia sclerotiarum, and then collar rot caused by Sclerotium ralphsi, leaf blight caused by stem phylum Sarciniforme, bacterial leaf blight caused by Xanthomonas campestris. Besides, some of the viral diseases like bean mosaic, bean common mosaic, alfalfa mosaic, and the stunt disease are very important in chickpea. Let us see every diseases in detail one by one. The Ascochyta blight, which is caused by Ascochyta rabi, which attacks all the aerial parts of the plants. The affected areas shows circular brown spots with grey center. At the center, there would be numerous black minute dots, which represents pycnidia of the fungi. The pycnidia are arranged in a concentric circle. In advanced stages, the blight area, which covers the major portion of leaf, and the entire leaf turn brown, scorchy, and completely dried. Similar type of deep-seated lesions may also occur on stem as well as on pods. So this deep-seated lesions on stem, which causes girdling symptom and cause collapse or wilting of entire plant. And similarly, the lesions on the pods, which causes shriveling of seeds, distortion as well as malformation of seeds. So the pathogen will produce brown septate mycelium and producing asexual fruiting body pycnidium. This is the typical shape of pycnidium, which in turn contains conidia, which are all oblong or oval, single-celled, occasionally bicelled in nature, and the high rainfall during the flowering stage as well as the temperature range of 20 to 25 degree with the more than 60% relative humidity favors the disease development. And then the pycnidia from the infected plant debris serves as source of inoculum. The pathogen may act both externally as well as internally seed-borne nature due to the infection on pods, and hence the infected seeds as well as the inoculum present in the plant debris serves as the primary source of inoculum, and then airborne conidia helps in the secondary spread of the disease. So to control this disease or to manage this disease, remove and destroy those plant debris which serves as the reservoir of diseases. And then if the disease is severe in the previous seasons, then better go for crop rotation with the non-host crop like cereals. So before sowing, treat the seeds with chemicals like thiram, carbendazim or thiram plus carbendazim combination fungicides at the rate of 2 gram per kg of seed. Exposing the seeds to a temperature of 40 to 50 degree, which reduces the pathogen about 40 to 70%. During the cropping period, you may also apply carbendazim at the rate of 500 gram per hectare to manage this disease. And then next one is wet root rot, which is caused by Rhizoctonia solani. The sexual stage is Thanatophorus cucumaris. Severe during the seedling stage of the crop. Dark brown lesions are formed at the base of the stem near the collar region at the ground level. The affected seedlings, they show yellow leaflets, drooping and complete drying of seedlings. The collar region showed rotting which turned black with black mycelial growth as well as with the sclerotial production. In the affected field, you can see the dead or dried seedlings due to the infection by Rhizoctonia solani. So the pathogen which produce dark brown mycelium and having constrictions in the hyphae at branching junctions, and the sclerotia it produces are dark brown and irregular in shape, heavy soil which is having poor drainage capacity, as well as warm humid weather are favoring the development of this disease. And the associated root rot, that is dry root rot, which is caused by Rhizoctonia bataticola, which is the sclerotial stage of the fungi. The pycnidial stage is mentioned as Macrophomina fasciolina. So this pathogen primarily causes seedling blight and collar rot in initial stages. In the grown up plant, the infection occurs during the flowering stage. The drying, dropping of leaves and death of plant that occurs in patches in the field. The shredding of barks at the basal portion and the shredding of roots due to the infection are the common symptoms produced by this pathogen, besides producing lot of sclerotia as well as pycnidia, dark brown color minute pycnidia at the basal portion of the infected stem. So this bataticola produces large number of round, irregular shaped sclerotia at the basal portion, and besides it also produces pycnidia with the characteristics osteole, which contains numerous single-celled, thin-walled, hyaline, elliptical pycnidiospores or conidia. So to manage this disease, before sowing, go for seed treatment with the chemicals like carbendazim or thiram at the rate of 2 gram per kg, or biocontrol agents like trichoderma at the rate of 4 gram per kg, or Pseudomonas fluorescens at the rate of 10 gram per kg is helpful in reducing the disease incidence. And basal application in the soil application with the biocontrol agents at the rate of 2.5 kg per hectare, along with the well decomposed farmyard manure at 30 days after sowing will be much helpful in the biological method of disease control. And among the chemical control, go for spot drenching with the carbendazim at the rate of 1 gram per liter of water and used for drenching purpose around the infected plants. Next important disease is rust disease caused by Euromyces ciceris aritini, so which produces brown rusty pustules on both the surface of the leaf. This iridosauri cover the entire leaf and leading to drying and defoliation. This iridosauri upon maturation converted into teleosauri. Similar type of raised brown pustules will also be produced on pods stem also. So the iridospores, which are single brownish in color, pedicellate and acuminated, while the teleospores are round, brown and single celled. So it survives as iridospores in the weed host Trigonella polycerata, and the repeating spore, that is iridospore, is responsible for severe spread of this disease. And to manage this disease, we can go for application of wettable sulfur at the rate of 1 gram per hectare, or carbendazim at the rate of 500 gram per hectare will be very good against the disease. And the next important disease is fusarium wilt. Fusarium wilt caused by Fusarium oxysporum, form of Specialis caesari. So it affects all stages of the crop, including seedling stage, flowering stage and then adult stage. In the seedling grown up plants, it produces yellowing of outer leaves and then followed by the younger leaves, and the drying of leaves and drooping of leaves are common phenomena in Fusarium wilt infection. And withering of plants at the later stage with brown discoloration in the vascular region. So browning of vascular region are also common phenomena. This vascular browning is prominent as black streak on the stem as well as internal portions of the stem and root. The pathogen produces hyaline, septate, highly branched mycelium, and the characteristic spores produced by Fusarium, which includes macroconidia, microconidia, and chlamydospores. The macroconidia are fusiform or sickle-shaped, multiseptate, while the microconidia are single-celled, oval-shaped, or spherical shaped, and chlamydospores which may be of terminal or intercalary. The high soil temperature and low soil moisture favors the development of this disease, and the weeds like Cyprus rotundus, Tribulus terrestris acting as an alternate host for this disease. So the infection on the lower leaves which causes yellowing and drying and drooping of lower leaves, followed by the younger leaves, and this is the typical vascular discoloration, dark black streaks due to infection by the fusarium. And macroconidia are sickle shaped, and then microconidia are single celled, oval or spherical shaped. And the seed borne as well as the pathogen survives in the plant debris, serves as the source of inoculum. The primary spread is mainly through the chlamydospores present in the infected plant debris, while the secondary spread is through irrigation water as well as through the culture during the cultural operations. So apply heavy dose of farmyard manure along with the green leaf manure, and soil application of biocontrol agents like Pseudomonas fluorescens or Trichoderma viridi at the rate of 2.5 kg per hectare, along with the well decomposed farmyard manure, which will helpful in reducing the soil borne inoculum. And then treat the seeds before sowing with the chemicals like carbendazim or thiram at the rate of 2 gram per kg, or along with the biocontrol agents like Trichoderma viridi at the rate of 4 gram per kg, or Pseudomonas fluorescens or Bacillus subtilis at the rate of 10 gram per kg. In the grown up plant in the field, as a chemical method, you can go for drenching with spot drenching with the carbendazim at the rate of 1 mixed at the rate of 1 gram per liter of water. Next important disease is collar rot, which is caused by Sclerotium ralphsi. So the name itself indicates it collar rot. It produces water-soaked black lesions near the collar region at the ground, just above the ground level on the seedlings up to 6 weeks. So infection at the basal portion that leads to yellowing and death of seedlings. The older seedlings after infection turn into dark, dried without collapsing, and also producing profuse mycelial growth, white mycelial growth, besides producing sclerotia. And then among the viral diseases, the chickpea stunt, which is caused by bean pea leaf roll virus, is very important. Which causes stunting of plant, shortening of internodes. So due to shortening of internodes, reduction in the leaf lamina are giving a bushy appearance of infected plants. So the leaves may turn yellow or with the orange discoloration, and ultimately leads to premature drying of plants due to the infection. And besides, it also produces browning or brown discoloration in the phloem region, especially on the stem near the collar region. And this virus, bean pea leaf roll virus, is transmitted by the aphid vector Apis toxivora as well as Mysis persicae. So to manage this disease, you have to rogue out the infected plant, which is largely advisable, or you can go for any one of the insecticides effective against the sucking pest. For example, spraying methyl demeton at the rate of 500 ml per hectare. And some of the fungal diseases are minor, fungal as well as bacterial diseases, which are minor nowadays, but that may become a major disease, after a long time, or after a period of time. So the leaf blight, which is caused by stem phylum Sarciniforme, this stem phylum is known to cause disease in coriaceous plants also. So this stem phylum Sarciniforme, which produces ovoid necrotic spots on the leaves with ashy borders. Besides, it also produces such necrotic spots on the stem and causes girdling of symptom and drying of leaves. Similarly, the bacterial leaf blight caused by Xanthomonas campestris, which produces small water-soaked lesions on the surface of the leaf later that turn into dark brown necrotic and surrounded with a chlorotic halo. And post-emergence seedling rot is also common due to bacterial this Xanthomonas campestris infection. And among the viral diseases, the bean common mosaic and alfalfa mosaic are serious on chickpea. The bean common mosaic, which produces stunted growth. So almost all the viral infections, the plant growth may be majority of the viral infections. The affected plant growth is retarded, so that would be stunted in their growth, and reduction or shortening of internodes, reduction in the leaf area that may give you bushy appearance. Besides, in the case of bean common mosaic virus infection on chickpea, it produces mosaic mottling pattern on the surface of young leaves. And this bean common mosaic virus is transmitted by the insect vector Apis gossypi or Apis craxivora. The same Apis gossypi as well as Apis craxivora also transmits the alfalfa mosaic virus, which produces twisting and twisting of the terminal bud followed by necrosis of affected portion, and due to necrosis initiation of secondary branches are occurring. These new branches will become very stiff, erect, with small crowded leaf and with mild mottling symptom. Thank you.